with the knowledge and with the sarcoma community realizing that there's no effective therapy for these patients, there's been several centers, such as my center, Sylvester Comprehensive Cancer Center, that have focused on research to try to find a treatment option for patients with chondrosarcoma. Another center led by Adrian Flanagan identified IDH1 and 2 mutations as a genetic abnormality that's found in over 50% of central chondrosarcomas. A pathologist at our center, in cooperation with Massachusetts General Hospital, identified isocitrate dehydrogenase 1 and 2, or IDH 1 and 2, as mutations that occurred in conventional chondrosarcoma, but not in chondroblastic osteosarcoma or other chondroid tumors, really. And so that led to the awareness that 50 to 75% of patients with chondrosarcoma, conventional chondrosarcoma, and dedifferentiated chondrosarcoma have mutations in one of these two genes, IDH1 or 2. These genes play a role in the normal cell energy metabolism. In other words, they play a role in a cycle called the TCA or Krebs cycle, where energy is made to make ATP so that normal cells in the body can survive and carry out their functions. Part of that process, IDH converts isocitrate to ketoglutarate, which is a normal process. It happens every day in all of the cells in our body. But once a mutant IDH is introduced into the cell, this enzyme converts the normal alpha ketoglutarate to a molecule that's normally not present in any cell in the body, called 2-hydroxyglutarate. And in fact, 2-hydroxyglutarate has been found to be one of the first so-called oncometabolites. And it converts for the normal ketoglutarate, it competes for the normal ketoglutarate for the binding to certain enzymes that prevent cancer. So when hydroxyglutarate binds to these enzymes, the ketoglutarate can't produce the tumor-causing processes. Thus, they take place, and these processes include DNA hypermethylation, histone hypermethylation, and gene expression driven by a protein called HIF1 alpha that's responsible for creating blood vessels in tumor cells. And all of this tumor phenotype is caused by the production by mutant IDH of this oncometabolite 2-hydroxyglutarate. So what investigators started doing is using an inhibitor of mutant IDH that doesn't affect the normal wild type IDH. This inhibitor called AGI 5198 is very potent, and the nanomolar range can shut down the mutant IDH and not affect the normal processes within the tumor cell. And so in our research lab, we have two cell lines that are conventional chondrosarcoma that both have IDH1 mutation. And when you incubate them with this inhibitor, you shut down the production of this oncometabolite 2-hydroxyglutarate, which is basically reversing the effects of the mutation in the tumor cell and the oncometabolite. One of my colleagues in the sarcoma world, Dr. Judith Bove, used this inhibitor to show that there was really minimal effect over the first few days when this inhibitor is incubated with the tumor cells that have IDH mutation. About that same time, some investigators studying brain tumors made the observation that if you incubated longer than that, like 6 months, you would start seeing reversal of the tumor phenotype thought to be related to differentiation, epigenetic differentiation, or reprogramming. So what we did was we took the same cell lines and we used this new technology called CRISPR, or gene editing, recently awarded Nobel Prize. And we're using this gene editing technology, we were able to basically cut out the mutant IDH genes in the cell line. So now we have 2 chondrosarcoma cell lines that have IDH mutation, and we have the same cell line that's exactly identical as each of those 2, except they don't have the mutant IDH. They only have the wild type IDH, 2 and 3, not 1. And so we studied these in long-term culture and showed that the 2-hydroxyglutarate, the oncometabolite that causes the chondrosarcoma, was shut down to essentially zero production. And then we looked at several different factors of the tumor cells, and mainly what was shut down was invasion and migration, not proliferation. We put the cells in an animal, put the tumor cells in mice, and the IDH mutant chondrosarcoma cells grew fine. They grew into these big tumors that would really affect the mice in adverse ways. However, when we put the exact same cell lines lacking mutant IDH, there were very, very small tumors growing very slowly. And so that was a signal to us that, in fact, mutant IDH was critical for chondrosarcoma tumor phenotype. So the next question was whether that could be carried over to patients. And so we then designed with several other chondrosarcoma experts around the United States, and in fact internationally, we designed a phase 1 study of a brand new mutant IDH inhibitor that was clinical grade, called Ivosidenib. In fact, that this new medicine called Ivosidenib, an mutant IDH inhibitor, was very well tolerated. Fewer than 5% of patients had any significant side effects, meaning 95% of patients really had no side effects from the medicine. And in fact, the patients that had conventional chondrosarcoma, over half of them were progression-free for 6 months. On the other hand, the patients that had the more aggressive chondrosarcoma, dedifferentiated chondrosarcoma, none of the patients were progression-free at 6 months. So we looked into the detailed tumor measurements and found that about 30% of the patients had some tumor shrinkage. About 30% of the patients had clear tumor growth. And then there were some patients that had a little bit of tumor growth but didn't meet the criteria for progression of disease. So it seemed that about 2/3 of the patients benefited from the therapy, either from slowing the growth of the tumor or outright shrinkage of the tumor. And we had a couple of patients that had really durable partial responses, not confirmed, but partial responses. And then we looked into a little more detail for the patients on the study. And what we found was that the conventional chondrosarcomas that had fairly straightforward genotypes, mostly IDH mutation only, did very well and met the criteria for stable disease or had some shrinkage of the tumor. However, if you looked at the chondrosarcoma patients that had the more aggressive dedifferentiated histology, or they had complex genotypes with multiple mutations in a number of other genes, such as retinoblastoma, P53, P10, RET, et cetera. These complicated, dedifferentiated tumors the patients didn't really seem to benefit from ivosidenib therapy. But clearly some patients did. So we went back to the cell line model to ask, why do some patients benefit and why do some patients not benefit? So in the cell line model, we found that knocking out mutant IDH, shutting down production of 2-hydroxyglutarate, led to epigenetic reprogramming and loss of expression of a category of proteins called integrins. Integrins are critical for invasion. They're critical for metastasis. They're critical for migration of tumor cells to other organs or into the blood supply. And so it seems that mutant IDH really drives this migration phenotype, invasion phenotype. But when other mutations take place, such as RB, P53, P10, RET, these other mutations then take over and really replace the tumorigenic phenotype that was initially started by IDH mutation. And so it's clear that we need to do more detailed epigenetic studies to determine the mechanism by which IDH mutation drives integrin activation and vasculogenesis. We're also very interested in exploring a potential registration study with an IDH inhibitor for our patients with chondrosarcoma, because they don't have any other treatment options. And even if a third of the patients benefit from therapy, we're at least helping some people that would have no other hope. We need better treatment options for these subset of patients that have the aggressive, dedifferentiated tumors, the complex genotypes. And it may be the fact that we need to use the IDH inhibitor in combination with other inhibitors or other anti-cancer therapies, such as trail analogs, such as tyrosine kinase inhibitors, such as even maybe chemotherapy. And to do this, we're going to need to do some in vitro drug sensitivity testing, which we can test hundreds and hundreds of new drugs in combination with IDH inhibitor. And we also may want to explore use of organoids or other in silico and in vivo testing to really find better therapies for these patients that really have limited treatment options. So thanks for giving me the opportunity to discuss our research and clinical trials and translational research in chondrosarcoma, studying IDH biology and therapy to try to bring treatments to patients that really have limited other options. Thank you.